Hello, my name is Kevin, and today I'll be presenting a research from the Lansky Lab at Michigan State University. This article is titled Genomic Evolution of Antibiotic Resistance is Contingent on Genetic Background Following a Long-Term Experiment with Escherichia coli. This study explored how genetic backgrounds can shape the way bacteria like E. coli evolve antibiotic resistant, even when they're exposed to the same drug in the same environment. This is the first page of the research article, published in the Proceeding of the National Academy of Science in 2021. This study was led by Kyle Card, Benjamin Good, and Jeffrey Berrick and Richard Lansky. As you'll see, it comes from a highly respected journal and a well-known lab that has been running a long-term evolution experiment with E. coli since 1988. E. coli is a common bacterium found in the intestines of humans and animals. While many strains are harmless, some can cause serious infections. The problem this study addresses is that E. coli doesn't evolve resistance to antibiotics the same way as across all strains. Even when exposed to the same drug, some strains become highly resistant while others don't. The author hypothesized that this variability is due to genetic background that is different in each strain evolutionary history. Their goal is to find whether prior mutations shape how resistance evolves, and whether we can predict which strains are more likely to become resistant. Antibiotic resistance is a major public concern. E. coli is known to evolve resistance quickly in lab settings. In some cases, evolution produces the same results, but in others it doesn't, raising the question, what causes that variation? One possible is genetic history, the background mutation already present in each strain before exposure to antibiotics. The researchers chose 12 E. coli strains, each with a unique genetic background from the long-term evolution experiment. They exposed all 12 to the same antibiotic, ceftriaxone, which is a beta-lactam. Over several days, they tracked how well the bacteria grew, how resistant they became, and which new mutations developed. They used whole genome sequencing to identify those genetic changes. The key result is that all 12 strains developed resistance, but not in the same way. Some strains became resistant much faster than others. Certain resistance mutations were only seen in specific backgrounds. Even when different strains got the same mutation, its effect on resistance varied depending on the genetic context. In the chart, shows how resistance levels changes across strains. On the name, you see different on the x-axis you see the different E. coli strains. In the y-axis shows the level of resistance measured as the minimum inhibitory concentration or MIC. As you can see, there is a wide range of outcomes, despite all strains being treated the same way. The main takeaway is that genetic background matters. It plays a huge role in determining how bacteria evolve resistance. Even with identical antibiotics and identical conditions, different E. coli strains took different evolutionary paths. One surprising finding was that the same resistant gene could be beneficial in one strain but harmful in the other. In the future, the researchers want to expand their work to other antibiotics and species. The goal is to better predict which bacteria strains are more likely to become resistant and why. Thank you very much. Hope you guys have a good day. Bye bye.